Hey guys, my name is Ashley Clark, and the paper that I chose for my long talk is Exomes derived from mesenchymal stem cells repair Parkinson's disease model by inducing autophagy. So the first thing we're going to take a look at is our outline. So we're going to go over a quick introduction, a few definitions, and then the fun stuff, so the paper itself, and then we just have a few takeaways at the end. So, about the paper, the overall purpose of this study was to actually establish a relationship between stem cells and neuronal development in the brain. And the main reason I actually chose this paper is because it's related to obstetrics. So, of course, we're going to go over this a little bit later, but the main stem cells actually used in this paper were obtained from human umbilical cords. So, the entire process was really fun to learn about, and I thought it would be a fun thing to talk about as well. I also decided to cover this paper mostly because stem cell treatments are pretty controversial, so I thought it would be something informative. So, Parkinson's disease. Parkinson's is one of the most frequent neurodegenerative disorders, and our little infographic on the left show a few motor and non-motor skill symptoms that come along with the prognosis of Parkinson's disease. So, something that I didn't know before reading this study was that the underlying mechanisms of Parkinson's are actually still pretty unclear, but researchers and scientists do believe that genetic and environmental factors are still pretty much involved. And the hallmark of Parkinson's disease is the loss of dopaminergic neurons, or just dopamine, in the substantia nigra. And the substantia nigra is a basal ganglia structure. It's located in the midbrain, and it plays a really important role in reward and movement. So, following the death of these dopamine neurons, a decline in dopamine metabolism, neuron inflammation, and mitochondrial function is almost always evident. And one of the two main changes we're going to talk about today is oxidative stress. And oxidative stress is the imbalance between the systemic creation of reactive oxygen and the system's ability to actually repair the damage that this process might have caused. And we're also going to talk about something called protein degradation or proteolysis. And proteolysis is just the breakdown of proteins into smaller polypeptides or amino acids. So, something to note, treatment cannot actually replace or replicate any dopaminergic neurons, but it does aim to decrease the severity of any symptoms that may be present in a Parkinson's disease patient. So, stem cells. Stem cells are, of course, already naturally produced by your body. They offer a self regulated regenerative response and limit the area of a lesion. So, a regenerative response is usually launched by injured organs or tissues and involves local populations of self renewing cells or the recruitment of actually already circulating stem cells to replace or repair any injured areas. And something else to note about stem cells that's super cool is something called differentiation. And differentiation means that stem cells are actually capable of basically transforming into any cell that naturally is already found in the body. So that's what our little graphic on the right here is showing. So we just see examples of different things that stem cells have the capabilities of turning into. So we're going to get a little bit more specific and talk about mesenchymal stem cells. So mesenchymal stem cells, or MSCs, are the most encouraging source for stem cell-based treatment. They have impeccable immunomodularity characteristics, proangionic features, and multi-lineage differentiation capabilities as well. So all of this is just to say that MSCs are the fixer-upper of stem cells. And MSCs can be isolated from multiple points on the body, which is what our graphic here is showing us, just different examples as to where MSCs can be isolated from. So, MSCs can be isolated from adipose tissue, bone marrow, dental pulp, and human umbilical cords via Wharton's jelly. And Wharton's jelly is the main MSC that we're going to be talking about today, and it's just a gelatinous substance, and it's found within the umbilical cords of mammals, and it's mostly made up of mucopolysaccharides as well as collagen and proteoglycans. So we're going to get a little more specific and a little bit smaller. So we're going to talk about something called extracellular vessels. And MSCs are actually comprised of encapsulated extracellular vessels as well as other solute factors. And EVs are actually nanosized, which is very small, And on average, they range from about 30 to 10,000 nanometers in diameter. And just for reference to visualize how small they actually are, a human hair is about 17 millionths of a meter, and 1 nanometer is actually 1 billionth of a meter. So, of course, a very big difference. So, EVs are membrane bound vessels. They're released from cells and they actually transport cargo among the body. So, cargo being DNA, RNA, and proteins. And EVs are actually used between cells as a form of intercellular communication. So, increasing new evidence actually indicates that EVs have a really strong impact on physiological processes and are particularly critical in cell to cell communication, especially in the brain. However, something to note, which I thought was really cool, but an excess of extracellular vessels can sometimes cause the progression of different types of cancerous brain tumors and even sometimes promote the growth of these cells. So we have to be very careful when we are using EVs when it comes to neurodegenerative disorders. So, the blood brain barrier. The blood brain barrier is to protect against circulating toxins or pathogens that could cause brain infections, but at the same time, it still allows vital nutrients to reach the brain. So, the blood brain barrier is really relevant and very important to note when it comes to nervous system diseases because it often makes it really difficult for drugs to actually reach the lesion site and have a therapeutic effect. However, just like how we talked about how small MSCs are, that means that they actually readily cross the blood brain barrier, which is very important. So now we're going to talk about HUC MSCs, so a little bit more specialized. HUC MSCs, or human umbilical cord stem cells, are one of the most favorite stem cells in stem cell treatment. Reasons being their multilineage differentiation capabilities, which we've already covered, and that again just means that they are able to change into a multitude of specialized cells that are usually created by tissue itself. But HUC MSCs also have something called autologous transplantation feasibility. An autologous transplantation is a procedure in which a patient's healthy stem cells are collected from the blood or bone marrow before treatment. They're then stored and then given back to the patient after treatment. So, this actually helps the body come back up to shape with the help of its own stem cells. And not a lot of stem cells can be taken out and replanted. So, that's really cool. A really cool fact about HUC MSCs. And human umbilical cord stem cells also have really easy acquisition and they have a lack of ethical problems compared to other stem cells that are used in stem cell research. However, it's only been seen that a small proportion of transplanted HUC MSCs have been actually found in target tissue because transplanted HUC MSCs have a really low survival rate in the host, and some studies have actually shown that vein transplanted cells may sometimes cause capillary immobilization and uncontrollable cell division. So, therefore, it's really important for scientists to keep on with the stem cell research because it's very important for us to be able to identify an alternative to actually replace HUC MSCs as a whole, but it still has a similar level of repair activity. So the pictures we're going to look at come directly from the study. The picture on the left is the morphological appearance of HUC MSCs. And this is actually the appearance of HUC MSCs at about 6 days of culturing. So after those 6 days are up, the researchers actually decided to replate the cultures. And after replating, the cells took on the appearance of long spindle-shaped fibroblastic cells and even began to form colonies. So that's what we're looking at in the right on the right side of the page. That's just a really zoomed in, of course, since we are looking at cells, but a really zoomed in picture of cells. So the last definition we're going to cover is autophagy. So autophagy is actually the most important mechanism that this entire experiment was based on, and it's your body's way of actually naturally cleaning out and reusing old cells, and we can actually see in our figure the entire process of autophagy within the body. So, autophagy, like I said, is a cellular mechanism and it recycles materials such as abnormal proteins and damaged organelles in order to maintain cell survival and homeostasis. So, a lot of researchers do believe that the alteration of autophagy may be a really key factor in contributing to the pathogenesis of Parkinson's disease. And a number of reports have also shown that increased autophagy has potential therapeutic effects in Parkinson's disease as well. So now to the fun stuff. Like I said, the article that I chose is Exomes derived from mesenchymal stem cells repair Parkinson's disease model by inducing autophagy. And this was published in a journal called Cell and Death Disease in 2020. So it's a pretty new paper. And in the study, it was basically investigated whether and how exomes repair the damage of Parkinson's disease models in vitro and in vivo by regulating autophagy in rat models. So the experimental design. This is actually a schematic that comes directly from the study, and I thought it was really important to add this just so it's easier to visualize every step of the experimental process. So in the very beginning, fresh umbilical cords were obtained from informed and of course consenting mothers and processed within about 4 to 6 hours of receiving them. So Wharton's jelly, of course, the MSC we were talking about previously, was extracted from these umbilical cords through the use of a cell culture medium. So the HUC MSCs were then left to culture in a serum-free environment, and then the culture medium was then collected with a centrifuge tube. So as we see in the beginning of our schematic on the left side, something called 6-OHDA was injected into the right striatum in order to actually establish an animal model of Parkinson's disease. So 6-OHDA is a synthetic compound and is often used by researchers to selectively destroy dopaminergic neurons in the brain. So it just allows researchers to actually study these neurodegenerative disorders without actually having an actual neurodegenerative patient in front of them. So, adult male rats were then anesthetized and injected with equal parts either 6-OHDA and saline or 6-OHDA and exomes. And there were about 12 rats in each group. And there was also something called a sham group, which was used as a control group. The words are pretty interchangeable. And that group was made up of 12 male rats as well. And those rats were injected with saline and ascorbic acid at the same dosages as the 6-OHDA and saline and 6-OHDA in exomes group. So the results. So following the administration of HUC MSCs, symptoms of dopaminergic neuron loss decreased on a physical and neurological level. So the HUC MSCs actually did end up crossing the blood-brain barrier, reach the substance nigra, decrease dopaminergic neurons loss, and improve something called apomorphine-induced asymmetric rotation. And of course, that's a really big word, and we're going to talk about it in the next slide. But overall, the HUC MSCs increase dopamine levels in the striatum, which is the most important part, of course. So the first graphics we're going to look at shows two different ways that the rat brains were actually assessed. So section A, which is on the far left, is the detection of tissue damage in the lesion side of the rat brain through a process called H&E staining. So on the top left, we have our sham group. The cells are pretty scattered, but they look pretty normal. In our 6-OHDA and saline group, we have really angulated, distorted, and honestly empty looking cells. They're not very healthy. And then in our 6-OHDA and exomes group, we have significant improvement and we see a lot more healthy cells. And just to better aid you in understanding what we're looking for, the more purple there is, that means that there's more healthy cells. So we want more purple. So the section labeled B, which is on the right, is the sections from each rat that were actually analyzed for the dopaminergic neuron counts by immunohistochemistry. So the images here show the 6-OHDA lesion side, which is the right side, compared with the unlesioned side, which is the left side. So the little brown dots that we're actually looking at are dopamine neurons and are actually seen to be a lot less scattered in the exomes group compared to the saline and sham group. And we also see that in the exomes group, the dopaminergic neurons are a lot more uniform, a lot more together, and even in size and in shape, they look a lot more healthy than the other groups. So next we're going to look at another test that the researchers used called a laser scanning microscopy. And in the laser scanning microscopy, brain tissue showed that the blue fluorescent intensity emitted by exomes was stronger and actually more localized within the substantia nigra in the rats. So on the top, we see these little blue dots, and the blue dots are just the HUC MSCs as a whole. And we see that in the 6-OHCA and HUC MSCs group, the blue is a lot more intense, it's a lot more together. So that means that the exomes actually did readily cross the blood-brain barrier, which is the most important part. And on the second graph on the bottom, this shows what we talked about in the last slide. So something called apopmorphine-induced asymmetric rotation. And apopmorphine-induced asymmetric rotation was assessed at weeks 4 and 8 after the first days of transplantation. And apopmorphine-induced asymmetric rotation was actually used in the animal model in order to mimic the motor responses of Parkinson's disease in humans. So, an apomorphine induced asymmetric rotation test is used to monitor the extent of motor impairment induced by the usage of 6-OHDA. So, compared with a 6-OHDA and saline group, animals injected with exomes showed a better regression and a lot less reductions in rotations over time compared with before transplantation and 4 weeks after. So all this to say that in the bottom graph, we see that the 6-OHDA and saline group, which is the line at the top, mostly stayed the same in terms of asymmetric apop rotation, and the 6-OHDA and exomes group actually declined significantly over time. So lastly, we're going to look to confirm the effect of HUC MSCs on dopaminergic neuron apoptosis, or just the diminishing of dopaminergic neurons in the brain. So to do this, scientists use something called a tunnel assay and a process called double labeling. So in double labeling, we're actually able to see every part of the brain, all the cells. So on the far left, we see the blue, so that's the nuclei of all the cells. In the box over, we see apoptosis via tunnel assay. This was highlighted by little arrows. And then dopaminergic neuron marker, that just shows us where the dopaminergic neurons actually are. So the results actually indicated that HUC MSCs actually protects dopaminergic neurons from 6-OHDA-induced apoptosis. So all this to say that the exomes actually were able to protect and lessen the effects of the 6-OHDA on the already there dopaminergic neurons. So, conclusion and next steps. So, overall, the use of MSCs to treat Parkinson's disease and a wide variety of other neurodegenerative disorders is pretty promising, which is, of course, good, even though the exact underlying mechanisms are still unknown. So, MSCs have become widely used for cell based therapy due to less scientific issues and ethical issues as well. So, MSCs are even more favored because of how small they are, like we talked about before. They're soluble in nature, and the most important thing is that they're able to cross the blood brain barrier. MSCs also cannot turn into anything harmful or malignant, which I thought was very interesting because a lot of stem cells are basically hard to control, but MSCs don't actually have the possibility or the capability of turning into anything that could be harmful for the body. And I also found that MSC exomes have also been reported to increase the neuroplasticity in the brains of rats with Alzheimer's. And they also reduce inflammation and increase neuronal regeneration in spinal cord injury and traumatic brain injury rats. However, further studies are still needed to identify specific approaches for other neurodegenerative diseases and to hopefully one day create a cell free therapeutic approach for neurodegenerative disease patients is the aim of stem cell research today. So, just a few takeaways. So, firstly, the underlying mechanisms of Parkinson's disease is still unknown, and thus that makes the disease actually uncurable since we are unable to localize where it actually comes from. but it is still treatable through the use of stem cells, which is the most important thing. Secondly, other stem cell treatments are urgently needed in order to avoid any further ethical and scientific issues. So that just means that because stem cell research is so under fire right now, it's really important for researchers to actually find another avenue to go down without the use of actual cells in treatments. And then, thirdly, although there are advances in the Parkinson's disease research field, I think it's really important to note that current therapeutic approaches improve Parkinson's disease patients' quality of life, but are actually not able to counteract the progression of Parkinson's disease nor stimulate any dopaminergic neuron survival or differentiation. So, this just means that we are unable to actually do better or reverse any progressions that have happened with Parkinson's disease patients, but we are able to better their quality of life. We're just aiming to significantly reduce the severity of any symptoms that they may be having. So, lastly, here are my references. And then I have a quick little meme at the end. Thank you.